I know, brevity is the soul of wit, and also a picture is worth 1,000 words, so I leave 20,000 words for you all, I thought speed is something that's important only in anchoring relay, but, it's here too, I guess, but I love speed, so let me skim through some of the things before I go into some of the experimental data that we have had in the last few years. And Dr. Pontis and Dr. Coley gave a nice cancer immunology 101, so it's much easier for me to skip some of the initial things. What is really needed for cancer immunotherapy is enlisting the body to fight cancer, obviously stimulating own immune system to work more harder as well as faster or smarter, and increase the specificity and amount of anti-cancer immunologic factors. There's not a single factor that is sufficient or single way of approaching it, and the personalized medicine is obviously going to add multiple paradigms to treating cancer based on immunotherapy approaches. This is a tumor microenvironment. Without going into the details, there are a lot of tumor promoting and tumor suppressive factors, and depending upon what activates more, it skews more towards the Th2 and impairs antigen presenting cell function, which is the fundamental concept of cancer being recognized. There are multiple ways you can optimize or stimulate the cancer fighting immune system. As you see, the first one is here you can just vaccinate, or you can make the cancer cells produce some of these DMCS, some of the factors which can stimulate and then irradiate and put inside. Or this is the dendritic cell, which I'm going to just talk a little bit about. Either you can get them outside and then pulse it with peptide and DNA or any of the vaccine, which will induce them to provide more efficient signal to the T cells, because the T cell and the dendritic cell connection is lost because of the tumor suppressing factors produced in the tumor microenvironment. The other thing is, can you also deliver these tumorigenic peptides or the signals directly inside a different target tissue where you can make the antigen presenting cells pick up those antigens and then efficiently interact with T cells. That's where I'm going to give you some outline of what we have been trying to do. This is once again Dr. Pontis described in addition to the lack of recognition or the antigen presenting cell function, recent studies have also demonstrated the presence or the role of CTLA4 or the programmed death receptor PD1 PDL interaction. These are beyond the T cell function normal, so the T cells do not function properly. There are monoclonal antibody therapies and other targeted therapies which are directly attacking towards this area, which are once again very powerful. And this is an outline of how we do cancer immunotherapy, which is especially ex vivo based. Isolate the progenitors and then differentiate, pulse them, put them back, and hopefully they fight better. So in addition to these sort of direct approaches on skewing or activating the dendritic cells, immunotherapy, cancer immunotherapy can also benefit in combination with multiple therapies, including chemotherapy, where you induce apoptosis of the cancer cells. What happens is there is cross-presentation. Once the cancer cell dies, obviously, the antigen-presenting cells can pick up any peptide or the signal that is given by the dying cell, and also you can combine that with GM-CSF other therapy. So personalized medicine comes very handy here, where most of these cancers which bear the tumor-associated antigens or some specific antigens based on mutations in the normal cell, the advantage there is, if there is a mutation which occurs only in the cancer cell population, you do not have the central tolerance. It's more a peripheral tolerance, so the immunotherapy can be much efficiently envisioned. And if you can sequence a person's DNA who has a cancer cell, and you can identify potentially some such peptides and develop tetramers to even isolate the T cells from their own body, or activate the T cells using the tetramer and the peptide that is identified, or using the TCR humanized mouse, where you can immunize these peptides and develop the T cell receptors, which can be used for autologous T cell therapy.